Hi, I'm Sandra Spurti. I'm a first year master student. The research work that I'm involved in is using In Cell NMR and cell differentiation technique to study neurodegenerative diseases. Now, what are neurodegenerative diseases? The examples for this are Parkinson, Alzheimer's, and dementia. There are many reasons for their cause, one of which is role of dopamine receptors. Especially Parkinson is second common disease, and the disease-modifying treatments are still not available in the market. Parkinson again is due to death of dopaminergic neurons in a part of our brain. Here, if we observe in the first picture, we can clearly see healthy neuronal cells releasing good amount of dopamine, whereas Parkinson affected neuronal cell is releasing very less dopamine. Hence, it is very important for us to study the neuronal cells that show positive activity for neurotransmitters. Now, it is again very difficult to find human dopaminergic neurons. Hence, there is a particular lineage called SHSY5I. This lineage has two cell lines, which is epithelial cell line and neuronal cell line. So these SHSY5I cells are taken and grown in regular nutrient media. If we observe the first picture is seen as a clump. This clump has both epithelial and neuronal cells. Once these cells are treated with the differentiating agents like retinoic acid and TPA, these cells, these RA and TPA, they help in good differentiation of axons and dendrites. We can even observe their communication with the neighboring neuronal cells. Now, these both differentiated and undifferentiated cells are taken for harvesting and casting. After that, they are taken for neuron NMR spectra. Now, in cell NMR analysis is again an emerging technique which has atomic level of resolution for metabolites that is occurring inside the cells. If we can observe the first plot it has two regions. One is outer region and one is inner region. The outer region shows one-dimensional proton peaks for buffer, glucose, lactate, alanine, and glutamine. Since these signals are overlapping, we can observe two-dimensional NMR spectra that is inside the first plot. So if we can observe glutamate, alanine, and lactate, only these metabolites are not overlapping in this plot. And the following peaks are the rates that are shown for these metabolites. Now, if we see first two plots, that is for lactate and alanine, they show steady state of peaks across the experiment, whereas glutamate reaches maximum level under controlled conditions. Such information at cellular level will pave a new way for drug testing and understanding pathogenic